My goal today, I think, was just to give you a broad overview of the Flavivirus DMS projects my lab has tackled thus far. Um, I want to give you an idea of the targets we've chosen and how we conducted those screens, and so a good amount of detail about the technical aspects of DMS. I'm only going to briefly touch on the downstream uses that we're pursuing with this data, sometimes because those are really like ongoing projects and we haven't figured a lot of those things out yet. So just situate, there we go. So my lab studies how flaviviruses co-evolve with their hosts, and we use a lot of molecular tools to do that. We make lots of mutations in both the virus and the host to kind of rack and check what the consequences are in each other, to try to understand how these viruses basically evolve functions to be able to infect hosts, and how the hosts have evolved the capacity to fight off viruses. Flaviviruses themselves are a quite large genus of different viruses. They're all enveloped, single-stranded, positive-strand RNA viruses. They all require a vector to transmit, and those are most often mosquitoes or ticks. And you can see a wide range of different, very famous viruses within this phylogenetic tree. The viruses I most commonly focus on are Zika virus and dengue virus, but we do a lot of cross comparisons between how those viruses function in respect to other viruses within this genus. Like I mentioned, our goal here is to talk about the deep mutational scanning that we've done. And so far for our studies involving DMS, we focused on the envelope glycoprotein, which I'll talk a bunch more about, and the NS5 or non-structural 5 protein of the virus, which is a methyltransferase and a polymerase. This is an obvious antiviral target. We're also working now with this AViD grant to do future DMS on the NS2B3 protease helicase protein, as well as the capsid protein of Zika virus. We want to expand that to dengue virus and West Nile virus as well, and help some of the other scientists do deep mutational scanning in their viruses of interest as well. One of the reasons we're interested in studying flaviviruses is that there really isn't a great vaccine available for any flavivirus, and there are no direct-acting antivirals as well. So it's really open to drug discovery. I want to give an early shout out to the person who taught me how to do deep mutational scanning, Jesse Bloom. So he is really one of the pioneers of this field, and he developed great ways to make libraries, to screen them and through complex systems, and probably most importantly, how to analyze the data as well. And so we did our first project pretty much hand in hand with Jesse, and now we're continuing to depend on him basically for at least the data analysis part of our projects. And he's been very kind and gracious at every step of these collaborations. So I'm very thankful for him for that help. But the basic premise for the DMS that we do is to make mutant viruses. And in this case, you make a library encoding all possible single amino acid changes in a gene of interest, and rescue a virus pool that thus would encode all of those single amino acid changes. And it's important to stress that these are saturating mutagenesis libraries, where every codon is changed to every other possible codon individually, and all possible mutations are represented in those libraries. If you screen that type of mutant virus pool in the right way, you can tease out, you can basically set up a competition assay where viruses are expanded directly proportional to their relative fitness. And if you deep sequence the rescue pool of passage viruses and compare that to the input pool of mutant viruses, you can get a very high resolution view of how a single amino acid change and how all single amino acid changes impact viral fitness. So here is the easiest way to look at this type of data, which is just a logo plot where the relative height of each letter at each codon position denotes its relative fitness. So a few things are really required for DMS to work in a virus setting. One is a highly efficient rescue system. So you need a plasmid that encodes a cDNA of a virus of interest that is very easy to clone in bacteria because you need to make large pools of of mutants in plasmids. And so we early on in the Zika virus outbreaks in 2016 developed our own infectious clone of one of the Zika virus MR766 strain of the virus. And this plasmid happened to satisfy those criteria. It was non-toxic in bacteria, which isn't true for all flavivirus encoding plasmids. And it produced large amounts of virus very quickly. And so what that meant is that we could generate lots of pools of mutant plasmids and rescue those efficiently and not bottleneck anything during those steps of our library screen. A few technical aspects about how we make those libraries are Jesse's old school way of doing this is to use overlapping PCR where sets of oligos are designed against a given gene where each codon has an opportunity to be essentially randomized through NNS mutagenesis. So you order a set of oligos for every codon you want to mutagenize. That's great. It turns out it works really well. And again, you just need to understand how many mutants you need to generate and how large a virus pool, plasmid pool you need to make sure that you've generated all possible mutants. Maybe the state of the art way of doing this now is to actually get synthetic DNA fragments where 20 different codons are present at each position. And there's a benefit there in these synthesized fragments that you have a slightly more even representation of the mutations in your given library. And it also means that you don't have to generate quite as large a library to represent every possible amino acid change. To be honest, I think we are still following the oligo overlapping PCR method for most of our studies, just because we have a lot of experience with it and we trust it. So the first project we tackled with the Bloom lab was to do deep mutational scanning of the Zika virus E protein. And there we were kind of just exploring the use of this technique in Zika virus for one thing, but we were trying to answer the question of the evolutionary dilemma where envelope proteins need to work. They need to be able to interact with host cells, but they also have to be able to tolerate mutations that allow them to escape from the adaptive immune response in a host. And so we want to understand the E protein's capacity to tolerate changes and what that flexibility meant in terms of antibody neutralization. So this was all published a few years ago, but just to summarize, we created a library of mutant plasmids, again, encoding all possible E protein single amino acid changes. You make sure it's large enough to overrepresent all possible changes. We rescued a mutant virus pool from that and passaged it at a low multiplicity of infection. That's to really lock the genotype and the phenotype relationship together. And then we conducted deep sequencing to calculate the relative fitness costs of each amino acid change. So this is a little bit of the validation data that we look at along the way. You can see, and we made actually 3 replicate plasmid libraries that were complete biologically independent, and we passaged them independently as well, and that gives us some feel for the reproducibility of our data. You can see that our plasmid libraries encode a large number of non-synonymous changes, stop codons, and synonymous changes as well. And when you passage those, you end up doing what you hope to do in terms of purging stop codons, which should absolutely be lethal for the virus, but especially in a virus like a flavivirus where this is a protein and a polyprotein, and any stop codon that you put in there will prevent any of the other viral proteins from being translated. We enriched synonymous mutations, which makes sense as well because those are the ones that evolved to be present in fit wild type virus. And we also selected a subset of non-synonymous variants as well. And of course, that's really the population that we were excited about analyzing. Just a quick look, a low resolution look at this data. You know, we expected that some regions would be highly conserved and other regions would be very flexible. So we looked at those regions first. And if you look at the likely to be highly conserved fusion loop, this is the part of the protein that actually catalyzes membrane fusion between the virus and the host membrane. You can see that the experimental, that the fitness, that the amino acid frequency variations in natural isolates shows it's highly conserved as it should be. And our deep mutational scanning data also showed that pretty much only the wild type amino acids worked in this position, except for one position that looks like it could tolerate a different amino acid. Now, we expected actually a region right under a glycan in the envelope to be very flexible, but this wasn't reflected in the sequence that was available from natural isolates. And that's pretty much just because there wasn't enough sequencing or variation in nature present at that time. But our DMS data does reveal a huge amount of flexibility within this glycan loop, where every single codon can tolerate multiple amino acids. And again, what's beautiful about this data is the height of each letter is directly proportional to its relative fitness. So that's a small look at the data. This is actually a big look at the data. All 504 or 5 amino acids of the E protein are represented here with the wild type sequence on the top line. And then every amino acid that can work at each position is listed below it. And you can get a pretty good feel that there are regions of high conservation and very low conservation. But in general, the wild type amino acid is the preferred amino acid, which shouldn't be a surprise since that's what's being selected naturally. And a lot of other amino acids can function. So right away, this kind of data is great because you don't have to test an individual amino acid to understand its potential fitness. You can just delve into this data set. Jesse has great algorithms to plot this flexibility onto protein structures. And so this is exhibiting that our DMS data on the E protein dimer. You can see it's actually 2 dimers kind of holding each other here with a fusion loop positioned just approximately in between the one end of the dimer and in the cleft of another. And again, the fusion loop is highly conserved here. And on this heat map, that's colored as very dark blue or purple colors. And so you can see that highly conserved fusion loop is present there. But in general, the surface of the E protein actually seems to be quite flexible. Most of the amino acids that are displayed on the top of the E protein demonstrate a considerable amount of flexibility. And maybe that's expected if those are the surfaces where antibodies are being targeted. If you look at the side view, there are regions that are more conserved or don't tolerate mutations as well. That are not present on the outside of a virus particle. So one of the great things about our DMS libraries, or DMS in general, is that you can use it to select for viruses that behave differently under different conditions. And one way would be to then select for viruses that are resistant to some type of inhibitor. And I think that's why this AViD grant is most interested in using DMS. But for this case, because we were studying the E protein, we wanted to study the ability to derive resistance to neutralizing antibodies. And the classical way to do that is to take a wild type virus stock and passage it in the presence of a given neutralizing antibody until a variant that is resistant to the antibody emerges and takes over the population during this passaging. And this is great. It definitely works to identify mutant viruses that are really resistant to neutralizing impacts. However, there are a bunch of limitations to this. One is it takes numerous passages to get to a resistant virus. You only really get one resistant virus out of a passage. and you can only get viruses that, or you're more likely to get viruses that are easily obtainable from a wild type sequence. So that's 1 or 2 nucleotide changes. You can't get all possible codons variants from that type of passage. So instead, we used our mutant library pools to select for all variants that are resistant to a neutralizing antibody. And we could do that in a single passage where we passaged a virus in the presence of a very high concentration of a neutralizing antibody, then which would result in only viruses that are resistant to that antibody surviving. And so that's shown in this overview on the top, where we passaged our mutant virus pools in the presence of a control antibody or 2 different monoclonal antibodies that neutralize Zika virus efficiently. I should say for at least one of these, we didn't know the epitope for that antibody, even though it was shown to be a very good antibody. So there was a discovery aspect to this as well. But what we did was we deep sequenced the viruses that survived that population. And to cut to the chase, here are the mutations that are differentially selected in the presence of those neutralizing antibodies. So these aren't adjacent amino acid positions. They're just positions that had a strong differential between a control antibody selection and either one of those neutralizing antibodies. What you get is a list of all possible amino acids that could confer resistance to an antibody. We tested that, and they do, in fact, result in neutralization resistance. What was really satisfying is, again, is, of course, these aren't directly adjacent amino acid positions, but if you plot them on the protein structure, they all cluster for the most part in very discrete locations, which we think highly define the epitope for that antibody. Of course, it's only the changes that can, the residues that can tolerate changes and provide resistance that we're really mapping here. And so there might be invariant residues that also form that epitope that aren't shown in that data. So we envisioned this kind of procedure to be done with any chemical inhibitor of virus replication as well. And then finally, what we're doing with the E protein is using our libraries to investigate how flaviviruses have evolved to be able to function in both insect and mammalian hosts. And to do that, we're passaging our viruses in parallel in cell lines representing those species, and then basically comparing the differential of how mutations are tolerated in one species versus the other. This is a dot plot of showing that many mutations have the same impact in both mammalian and insect cells, but a lot of mutations fall off of that diagonal, and we hope that these are species-specific mutations, which we're still testing, so we don't know for sure. So the second target that my lab has taken a DMS view of is the Zika virus NS5 protein, which is an enzyme. It has RNA-dependent RNA polymerase activity in this region, and a methyltransferase in this, and these are essential to viral replication. My lab got interested in this because we wanted to study how those activities overlapped with a third activity, which is to antagonize interferon signaling. So this protein also binds to human STAT2 and induces its degradation, and that's critical for in vivo infection and pathogenesis. And we wanted to understand how these 3 activities co-evolved on the same protein. So to do this, we actually were a little intimidated by the size of the NS5 protein. It's 903 amino acids, and we didn't think we could avoid bottlenecks if we did a DMS of that size of protein all at once. So instead, we broke it up into 110 amino acid tiles, which basically represent a good deep sequencing amplicon. And what we were thrilled to see is the data we generated with that tiled effect was really great, where our biological replicates had strong correlations with each other. Just to show you what could go wrong is our E protein DMS biological replicates weren't nearly as tight, and I don't think I fully appreciated that the evidence of bottlenecks when we did that first project. So this data is really amazing. That's the logo plot of the full NS5 protein, and it's not really worth looking at that. Instead, it's better to look at it plotted on the protein structure, where what we see are regions of high conservation where they can only a few, one or two different amino acids can function, and regions of high flexibility as well. Of course, that's what we expect. We're actually a little surprised that large faces of this protein are so highly conserved. If you look at things that we know are important about the enzymatic functions, the polymerase active site is absolutely conserved, which you would expect, as well as a priming loop that's important for the RNA synthesis functions. What we knew here was that a region of the priming loop should be conserved, but we were interested to see that a large amount of flexibility leads up to that conserved region within this loop, and you can trace that in these orange outlined amino acids here. So those all make sense to us. We, of course, validate our DMS data, and for this, we just clone a bunch of mutants and test their actual fitness. Here, we're using a luciferase virus to test the actual fitness of all of these mutants, and we plot that against the predicted DMS fitness. And what you see is a good correlation where you're able to score viruses that are near dead or likely to be dead, and those that are alive with some resolution within the alive space as well. But for us, it's mostly important for us to be able to call what's a dead amino acid change and what's a live amino acid change. So our current goals for the Zika virus NS5 DMS are to, again, define the overlap between the replication and interferon signaling antagonism functions of this protein. We're still working out how to do interferon antagonism DNS, but we've done a lot of mutagenesis to be able to really understand how these functions are overlapping. And so we're still working on that. We want to do a similar insect and mammalian cell screen of our DMS libraries to look for NS5 species-specific replication determinants. And of course, now with the AViD grant, we're interested in mapping how mutations can provide resistance to inhibitors. The other targets that we're interested in, again, are like the capsid protein and the NS2B3 protease are definitely in the same sort of pipeline as that. So I hope I gave you a good overview of how we use DMS to study Flaviviruses. We determine the evolutionary capacity to tolerate changes. We use these libraries to map resistance to inhibitors, and we're studying things like species-specific mutational impacts as well. And so I'd like to thank my lab, particularly Blake and Marion, who carried out the NS5 and E protein DMS. And none of this would have been possible without the help of Jesse Bloom and other people in his lab as well. And so I'm very grateful to them. So thank you.